actually, Dr. Rose's group already thought of the existence of such a modifying factor back in 1998, and they started looking for cis-acting, so variations, genetic variations that work in concert with the ApoE alleles, and in what we call the linkage to Cebioglu region. And in the last several years, there have been many genome-wide association studies generated for Alzheimer's disease, over a dozen of them. And all of them, of course, show replicated association for the ApoE region. It's two different variants define the ApoE 2, 3, and 4 variants. But all these studies also identified some key variants in the TOM40 gene, which is adjacent to ApoE. And you can see this astronomical p-value of 1 point at the top there, 1.8 e minus 157. So clearly there is something going on there. And the questions, and so we were wondering what it is it independent, how does it interact with ApoE4, how do we, and what does it do biologically, of course. So we used a tool that has been rarely, if at all, used for human genetics before we used it. It's called Phylogenetics. It was first developed for viral mutation analysis. This is actually how vaccinations are developed yearly. You look at the new strains of viruses and their relationship, and then you produce a vaccine accordingly. And what it does is it can analyze simultaneously different mutations or different variants that are located closely on the same sequence without the need to go SNP by SNP or marker by marker, which is the usual way we go about doing genetic analysis. And another advantage is that you're not constrained to only look at SNPs. You can look at any type of variation, allele repeats, insertion deletions, whatever the variant is, you can put it into the analysis. And the idea is that it takes into consideration the evolutionary model that led to the creation of, or to the elicitation of these mutations or these markers. So it's called the Coalescence, where you look at genetic variants that appear today in the genome and how did they come about to accumulate one on top of the other. And this is one example from two different papers on how you can look at those different variations and create those, or start putting the models on the evolution of that was behind the creation of the sequence on each specific molecule or each specific chromosome. And this is that region. You can see TOM40 and ApoE at the very top. That's the sequence. And these are three different windows we looked at. So we used Sanger sequencing, so it was very important to use deep primary sequencing, because all these novel next generation sequencing methods, they are practically useless when it comes to triple repeats or homopolymers or any small structural variations. Technically these methods cannot sequence them right, so you have to use Sanger sequencing. And that's what we did here. And we did the phylogenetic analysis for each one of these bins, and we found a very good bootstrap support, so statistical support for one of them, and we focused on that. Just kind of to give you an idea of what the genetic variation in that gene looks, in that region looks like. So these are the 6 axons on the POM40 gene. You can see all these straight lines are SNPs. It's a very variable region, and this is pretty common for any gene, a lot of variation there. And then there are 4 boxes of poly-T repeats. And just to, another clip, you can see the difference between a SNP and a poly-T. So a SNP on the very left there is when you have one nucleotide difference, a G or a A or something else, but it's a single nucleotide. When you look at the poly-T repeat or a homopolymer polymorphism, you can see that there is one nucleotide that keeps on going, and the number of nucleotides can be highly variable. So in this case, you have a genotype, so 2 chromosomes that a single person carries. One of them can have 16 Ts, the other 25. So it's a very big range. And actually, for this specific, the 523 variant that we're going to focus on today, can range between 14 and over 35 Ts. So it's a huge variability, and as you might expect, it has biological and clinical relevance. In the next slide, I'm going to go through very quickly the initial findings. We looked at a cohort of 105 patients with AD, and we found that this marker over there, it's a SNP, differentiated between these 2 bins, and another click will show that even though this is a SNP in POM40, you can see that it already categorizes the ApoE carriage between patients quite significantly. In particular, if you look at the ApoE4 carriers, which we said already are the ones that will develop Alzheimer's at an earlier age, all of them are bent in A, none of them in 2B. Another click. You can see that there is enrichment for case control, so many more cases in A than in B, which is expected because the ApoE4 fours are in A. Now, when we looked at these, so when the edges, those pancake figures that we see at the very edge of these tree structures are actually the same variant. They're all the POM40, 523 markers of poly-T I showed you, all of them. And what happens is that each one of these boxes actually is a distinct range of poly-T length. And 2 more clicks, and you can see that the short version is only in B. These are the ones that have favorable outcome and actually later disease onset. And all the others, the very longs and the longs, are on A, and they are prone to have disease early on.